Most of the molecules in the foods we eat are organic, and they contain many of the functional groups we discussed earlier. Monosaccharides are a group of organic molecules that living organisms use to produce energy. Monosaccharides are simple sugars with a generic formula. While there are hundreds of isomers with the formula C6H12O6, one of them is found in all living cells, glucose. Because carbon forms tetrahedral arrangements of atoms, the molecule isn't really straight. It looks more like the image shown here. Glucose contains aldehyde and alcohol functional groups. Sometimes, one of the alcohol groups reacts with the aldehyde to produce a cyclic molecule, where some of the atoms are connected in a ring. When that happens, we get either a molecule where the alcohol group on carbon 1 points down, called alpha-glucose, or a molecule called beta-glucose, where the alcohol group is structurally similar to another important hexose, fructose. Fructose is different from glucose because it has a ketone group rather than an aldehyde group. Because of its different structure, fructose forms a cyclic structure with 5 atoms in the ring. Glucose and fructose are important hexoses. Ribose and deoxyribose are the most important pentoses. Both pentoses contain an aldehyde group, and they form 5-membered ring structures. The main difference is at carbon 2. Ribose has an alcohol group here, while deoxyribose has just a hydrogen atom. A tiny variation of just a couple of atoms makes all the difference in the world. Deoxyribose is found in deoxyribonucleic acids, or DNA, our genetic material, while ribose is found in a fatty acids are one of the most important forms of energy storage in living organisms. A fatty acid is a long hydrocarbon chain with a carboxylic acid group at one end. The nonpolar hydrocarbon part of the molecule dominates its chemical behavior, making it hydrophobic. Some fatty acids are saturated. This means that the carbon atoms in the hydrocarbon chain form single bonds to hydrogen atoms and to other carbon atoms. Unsaturated fatty acids, like linoleic acid, have one or more carbon-carbon double bonds. These carbons are not yet saturated with hydrogen atoms. Fatty acids with one double bond are called monounsaturated, and fatty acids like linoleic acid with more than one double bond are called polyunsaturated. Unsaturated fatty acids are considered healthier than saturated fatty acids. Cholesterol is an important molecule for the digestion of fats, even though it's not used for energy itself. Cholesterol is based on the ring structure shown here. This collection of 4 rings is called a steroid ring structure and is found in many steroid hormones such as testosterone and estrogen. All steroid compounds have this structure, but they differ in the functional groups found on the ring. For example, cholesterol has an alcohol group on one ring, a double bond in another, and a hydrocarbon group on the 5-membered ring. Animals, including humans, synthesize cholesterol from the breakdown products of fatty acids. Cholesterol is the starting material for many other biological molecules, including bile salts, which aid in the digestion of fats. A diet high in cholesterol, like a diet high in saturated, let's look more closely at 2 groups of biologically important compounds, amino acids and nucleotides, whose structures include the nitrogen-containing functional groups. Amino acids contain an amine functional group and a carboxylic acid group. Under most biological conditions, amino acids exist as zwitterions. It's helpful to classify amino acids according to their side chain structure. Some amino acids have a nonpolar side chain. Alanine is one example. The hydrocarbon side chain, CH3, is what makes this structure nonpolar. 8 other biological amino acids also have nonpolar side chains. If one of the hydrogen atoms on alanine's side chain is replaced by an alcohol group, we get the amino acid serine. The alcohol group makes the side chain polar because the electrons in the carbon-oxygen bond are closer to the oxygen. 5 other biological amino acids also have polar side chains. Finally, some amino acids have ionic side chains. If one of the hydrogen atoms on alanine side chain is replaced by a carboxylic acid group, we get aspartic acid. The names and structures of all 20 amino acids are presented on the reference sheet that accompanies this activity. The structure of an amino acid side chain influences its physical properties, but it also determines its chemical reactivity. Nucleotides are the other nitrogen-containing biomolecules we'll consider. Nucleotides have three components: a nitrogenous base, a pentose sugar, and a phosphate group. When the structure consists of only the can classify nucleotides according to the pentose sugar they contain. Nucleotides containing the pentose sugar ribose are called ribonucleotides, while those containing nucleotides use one of 5 different nitrogenous bases. 2 of the bases, adenine and guanine, are called purines. They're found in ribonucleotides and deoxyribonucleotides. The other 3 bases are pyrimidines. Cytosine is found in both ribonucleotides and deoxyribonucleotides. Uracil is found only in ribonucleotides, and thymine is found only in deoxyribonucleotides.